Hello my dear friends, welcome to Live Prep. Myself Dr. Gajapati Verma. As I informed you earlier, under the name of Bootcamp, we are providing both revision lectures and also test series. And these revision lectures are available both on Live Prep app and also on our YouTube channel. But this test series can be given only through Live Prep app. Now with this, now let us quickly go with the revision of the chapter that is Biomolecules. In the previous lecture, we have studied almost half of the biomolecules chapter, and there we have covered about the basic introduction related to chemical analysis. And then under the molecules, if we are specifically talking, we studied about amino acids, proteins, lipids, nucleotides and the nucleic acids. Up to that we have studied. Now in this lecture, we will cover the carbohydrates, primary metabolites, secondary metabolites. Very small information is there in NCERT, and then directly we are going with some enzymes related information. Clear? With this chapter will be completed. Let us quickly start with this one. And here we are talking about the carbohydrates. So generally when we are talking about carbohydrates, in the name we are talking that carbon hydrates. So hydrates of the carbon. So but technically they are not hydrates of carbon. So in the molecular formula, if you are analyzing, yes, you can write it as hydrates of carbon, but they are not hydrates of carbon. Clear? And we are classifying them into monosaccharides, oligosaccharides and then polysaccharides. Clear? So monosaccharides, they cannot be hydrolyzed further, so that's why they are the simplest units. But in the range of 2 to 10 units, if they are present, generally they are considered as oligosaccharides. So within the oligosaccharides, we write it as disaccharides, trisaccharides, tetrasaccharides, pentasaccharides. Likewise we are writing. And then finally, if more than 10 are present, now generally they are considered as polysaccharides. Poly means many sugars are present. Saccharides means sugars. So many sugar units are present. So in this way we are writing this general classification. So in this, now slowly we will go with each of these. So they are actually, carbohydrates are actually polyhydroxy aldehydes or ketones. Either they are having aldehyde group or ketone group. One of this will be compulsory. Along with this, many hydroxy, many OH groups are present. So that's why many OH group containing aldehydes or many OH group containing ketones. So these are the carbohydrates. Clear? And here we are classifying them into monosaccharides, oligosaccharides and polysaccharides. One by one we will slowly go through this. Monosaccharides, single, mono is the single, right? So saccharides are the sugars. So these are the single sugars. These are the simplest sugars. These cannot be hydrolyzed further. So that's why these are called as monosaccharides. Either aldehyde group or ketone group is present. Those sugars which are having aldehyde group are called as aldoses. Those sugars which are having ketone group are called as ketoses. So this is how we are writing the name. Next one, how many carbons are present in this? So based on this, if 3 carbons are present, trioses, 4 carbon tetroses, 5 carbon pentoses, 6 carbon hexoses and 7 carbon are called as heptoses. So in this way different names we are using. And now this is the list. So 3 carbon triose having aldehyde group. So triose and aldose. So what are the examples, important examples? Glyceraldehyde. Standard examples we are writing. So ketone, dihydroxyacetone. So 4 carbon aldehyde group erythrose, ketone group erythrulose. 5 carbon aldehyde group ribose, xylose, arabinose. Not only ribose, 2-deoxyribose. That is also standard example, right? That is also part of 5 carbon containing aldehyde sugar. Next one, under the ketoses we are talking about ribulose and xylulose. Next one, under the 6 carbon, important examples are glucose, galactose, mannose. These are all aldose sugars. Next one, fructose is ketose sugar. Next one, regarding the heptose. So aldehyde group containing glucoheptose and galactoheptose. So regarding this ketone, if you are talking pseudoheptulose. So these are some standard and important examples from this monosaccharides. Not only this, again in each group, so many other examples are also present, but they are all not needed, but standard some important examples, please go through. But please remember, even though in the biomolecules chapter monosaccharide, disaccharide information was not given, still at least some basic information you need to remember. And majority, please remember, majority of the monosaccharides will follow the formula called as CnH2nOn or CnH2ON times. Like that also you can write. But please remember, some monosaccharides, they are not following this formula, but still they are considered as monosaccharides. Whereas some other molecules, they are following this formula, but they are not monosaccharides. In this way, different exceptions are also present. So that's why we are saying that majority of the monosaccharides are following this particular formula. Clear? Next one, important pentoses. What are the various important pentoses? Ribose sugar. Where we are studying the ribose sugar? Ribose sugar is part of the ribonucleotides. The nucleotides which are forming RNA, they are generally having this, not generally, they are always having this ribose sugar. Whereas 2-deoxyribose, this is also a pentose sugar, and this sugar is present in nucleotides that are part of DNA. So that's why these are some standard and important examples. Next one regarding the 6 carbon containing hexoaldoges, 6 carbon containing aldehyde containing sugars. What are the 3 standard examples? Glucose, galactose and mannose. 3 important examples. Out of this also we are talking about epimers examples. So here glucose and mannose are second carbon epimers, whereas glucose and galactose are fourth carbon epimers. This concept with more details we studied in the regular lecture. So if you are having any doubts, so please go through the regular lecture. Next one here regarding the glucose, if you are talking here also. So this glucose is called as the blood glucose, blood sugar. Yes or no? And next one, major form of the energy source is the glucose. That's why whenever our body requires the energy, majorly it is depending on the glucose. And our body, so excess food is stored in the form of glycogen. That glycogen is again polymer of glucose. So glycogen will break down into glucose. Now this glucose will be available to the cells. Now cells will try to get energy from this particular glucose. Not only this, some other compounds also by interconversion, they can also finally give energy, but this is the major form. Clear? Now here also that epimer concept, if you are not epimer, anomer concept, that alpha glucose, beta glucose, likewise different structures are also possible. So now these structures we are talking with respect to the carbon where this aldehyde or ketone group are present. So there we are actually talking this. So more details we have discussed in the regular lecture. So please go through that one. Next one is regarding the fructose. So fructose is 6 carbon containing ketose sugar, and here it is also called as a fruit sugar because a majority of the fruits they are having this fructose. So that's why fructose is called as a fruit sugar. But since it is called as fruit sugar, some students are making mistake here. Observe carefully. Suppose they are asking that fruits contain which of the following sugar. So here glucose, fructose, sucrose, likewise they are giving all of these, they are giving, suppose all of these they are giving, then you should go for all of these. Please remember, major form of the sugar is the fructose. It does not mean that only fructose is present. Some students are thinking that since fructose is called as fruit sugar, fruits will have only fructose. They are thinking like this. That is wrong. That is wrong. Other forms of sugars are also present, but the major form is fructose. This is what the meaning is. So try to understand the correct meaning of these sentences. Next one. So in the honey also different types of sugars are present. So fructose is also one of them. This is what the meaning is. Next one. In the seminal fluid, so in the human reproduction chapter you have studied this. The seminal fluid is also having the fructose, and this fructose is used as energy source for the sperm cells motility. Yes or no? So that point also we have studied. So please interlink all these informations. Clear? So these are some important examples for monosaccharides. Now we will talk about the oligosaccharides. Oligosaccharides generally they are having 2 to 10 units. And here if they are having 2 units, they are called as disaccharides. In the disaccharides again 3 important examples we are studying. First one is the sucrose. In the sucrose what are present? Glucose and fructose. And both the carbonyl carbons are directly attached with galactoside bond. Sorry, in general we are talking this one as, so glycosidic bond, not galactoside, glycosidic bond is present between these. Okay, so carbon 1 of this, carbon 2 of this, both of them are involved here. That's why it is glycosidic bond. And please remember here carbonyl carbon is not free. In the case of sucrose, carbonyl carbon is not free. That's why it is non-reducing sugar. Very, very important example. Sucrose is also called as invert sugar. It is an example for non-reducing sugar. Very important. Remember this. Next one regarding the lactose. Lactose is also called as milk sugar. Okay, it is called as milk sugar. In this what are present? Galactose is present and glucose is present. But this is having free carbonyl group. So that's why it is example for reducing sugar. Next one is regarding maltose. It is also called as malt sugar. It is having both the units are glucose units only. And both the glucose units are linked by glycosidic bond. And here also free carbonyl group is present. That's why it is example for reducing sugar. Clear? These are some important points you need to remember. Then what is reducing and what is non-reducing, and how we will determine this. Please remember, if a sugar is having free carbonyl group, either aldehyde group free or ketone group free. A free carbonyl group is present, that is reducing sugar. If a free carbonyl group is not present, that is non-reducing sugar. That's why all the monosaccharides are reducing sugars. Within the disaccharides, sucrose is non-reducing, whereas lactose and maltose are reducing sugars. Please remember this information. Very, very important for examination. Clear? These are some important points that you need to remember. Next one regarding the polysaccharides. Anyhow, in NCERT also they have mentioned about polysaccharides. So these are macromolecules, large molecules. In the macromolecular fraction, in the acid insoluble fraction, we are getting these polysaccharides. Where will you find these monosaccharides and disaccharides? Mono and disaccharides, they will come under acid soluble pole. In the filtrate fraction, we will get them. But these polysaccharides, we will find them in retentate. In the retentate fraction, we will observe this. Next one, these are long chains of sugars. Many sugar molecules are attached to each other. That's why they are forming this polysaccharides. And here in NCERT they are mentioning about some important examples. And here cellulose. Cellulose is a polymer of glucose. That means many glucose units are attached to each other. And here the glucose is present in beta anomeric form. So beta glucose units are attached here. And here, since all these are beta glucose only, so you can call cellulose as a homopolymer because all of them are of same units and it is a homopolymer. Next one we are talking is regarding the starch. Starch is also polymer of glucose, but in the case of starch, glucose is present in alpha form. But since all these are alpha glucose only, you can say that starch is also homopolymer. But starch is the storage polysaccharide. It is a storage polysaccharide. Food is stored in the form of starch. In which organisms? In the case of plants. Please remember this. Next one is regarding the glycogen. So in the case of glycogen, here it is also having alpha glucose. So now you need to understand the difference between starch and the glycogen. Starch is also having alpha glucose. Glycogen is also having the alpha glucose. Then what is the major difference here? It is about the branching. More branching is seen in the case of glycogen. That's it. So that's why glycogen is also called as animal starch. Glycogen is also called as animal starch. In the case of animals, food is stored in the form of glycogen. If you are talking with respect to the human, so human are also example under the animals. So in the case of human, energy, food is stored in the form of glycogen. Glycogen in the skeletal muscles, glycogen in liver. Please remember this. This is very, very important for examination. Clear? So this is also homopolymer. Next one is regarding the inulin. Spelling is very, very important. It is INULIN, inulin. It is not insulin. Insulin is a protein which is acting as a hormone. But inulin is not a protein. Inulin is a polysaccharide and it is made up of fructose. It is made up of fructose and it is a storage polysaccharide in some tubers like dahlia. So it is a storage polysaccharide. So please remember this. Okay, and here regarding the inulin, if human eats the inulin, we cannot digest the inulin. We cannot digest the inulin because we don't have the enzymes to digest inulin. So please remember this. So we cannot digest cellulose, we cannot digest inulin. But we can digest starch and we can digest glycogen. Please remember this. These are some important points. So all these four examples, so cellulose, starch, glycogen, inulin, all these are examples for homopolysaccharides. All these are having same type of unit. Though they are different in each type, within a specific type, all the units are same. That's why they are examples for homopolysaccharides. Next one, in the NCERT they have given this type of diagram, and please remember when you are facing this is the left side end, this is the right side end. This is the standard convention. Left side end is called as a non-reducing end, whereas the right side end is called as reducing end. Why it is like this? Because in the standard way of writing, first unit will be written on the left side, next one is on the right side, next one right side, right side, right side. In this way we are writing. When we are writing like this, now at the right side end what happens? Now free carbonyl group is present. That's why it is reducing end. But here free carbonyl group is not present. It is already in the bonding. So that's why left side end is called as non-reducing end. So please remember this. And overall if you see, polysaccharides are considered as a non-reducing. And please remember the term sugar. The term sugar means sweet, but the term sugar is not applicable to polysaccharides because polysaccharides are not sweet to taste. They are not sweet to taste. So these are some important points you need to remember. Since we are talking about this left side and right side end, there is some interlinking information, so that I will tell you. So please remember this. Now whatever we are studying, it is based on international standard convention. So everyone should follow this same convention only. Please remember this. Suppose I am drawing a particular line. So for this line, as per international standard convention, so now this is the left side, and this is the right side. Now for different types of molecules, what type of convention we are using? Observe this carefully. It is interlinked information. Observe carefully. Suppose you are talking about DNA molecule. If you are talking about DNA molecule, for DNA molecule, what will be the left side end? For DNA molecule, left side end is 5 prime and the right side end is 3 prime. Right side end is 3 prime. Left end and right side end. Observe carefully. Next one, if you are talking for RNA molecule, for the RNA molecule, suppose this is RNA molecule. For the RNA molecule, what is the left side end? So as per convention, left side end is 5 prime end, right side end is 3 prime end. That's why always reading of nucleic acids, whether it is DNA or RNA, whatever it is, the reading of these nucleic acids is always from 5 prime to 3 prime only. This is the standard fixed sequence. Please remember this. This is about the nucleic acids. Then we will talk about the protein. Regarding the protein, in the previous lecture itself we studied. So suppose this is the standard sequence. Now what is the left side end is called as? From which side to which side we need to read? So left side end is called as N terminus, amino terminus, whereas the right side end is called as C terminus, carboxy terminus. These are standard conventions. So please remember this. Suppose you are talking for a polysaccharide. Suppose we are talking for a polysaccharide. For the polysaccharide, just now we studied. What is the left side end? What is the standard sequence here? Standard information here. Left side end is non-reducing end. Whereas right side end is reducing end. Please remember this. Don't ever make any mistake. For both the nucleic acids, always the reading is from 5 prime to 3 prime. And for protein, the sequence is from N terminus to C terminus. For the polysaccharide, it is from non-reducing end to reducing end. This is how you need to talk whenever you are talking with respect to standard convention. The same information they have mentioned in NCERT. This is what they are talking. So this is what the interlinking that you need to know. What is standard? What is conventional? All these things you need to understand. Next one, they are talking about the starch. Starch is having helical secondary structures, and in these helical secondary structures, it can hold the iodine. So since it is holding the iodine, so it is giving blue color when iodine is interacting with the starch. So you will find blue color when iodine interacts with the starch, because this iodine is held within the secondary helical structures of this starch, whereas cellulose does not form these helical structures. Since cellulose is not forming helical structures, it cannot hold iodine. So that's why it cannot form blue color. So these are some experimental things. So please try to understand this. Clear? Since we are talking about these helical structures, now I will interlink another important information. So what is that important information? Now let us see here. Just now we studied about helical structures, helices of starch. Just now we studied about helices of starch. Nice. Next one, in the previous lecture we studied about alpha helix of protein. Alpha helix of protein we studied in previous lecture. Yes or no? Next one, we studied another helix, that is collagen triple helix. Collagen triple helix. Please interlink the information so that it will be easy to remember. Collagen triple helix. And for DNA, when we studied about double helical structure of DNA. Double helical structure of DNA. It is also called as BDNA model. Watson Crick DNA model. Yes or no? So in this way we studied about different helical structures. So here the helical structure for the starch, alpha helices of protein, collagen triple helix, and DNA double helix. So what is common point in all these things? All these are secondary structures. Please interlink the information so that you can easily understand the concept. All these are secondary structures. So this is secondary structure. This is also secondary structure. This is also secondary structure. This is also secondary structure. So in this way, in the examination, they can give some sentences so that you should not make any mistake. So please understand the concept. Don't try to by heart this. So this is what the interlinked information. So with this, that part is regarding that starch is completed. Now we will talk about the cellulose. Just now we studied that cellulose cannot hold iodine because it is not having helical structure, and the cellulose is generally seen in the cell walls of plant cells. And then, so whatever the paper that we are using and whatever the cotton that we are using, so all these are made up of, again, mainly consist of cellulose only. So some examples they gave in NCERT, at least NCERT examples, please remember. Next one is regarding complex polysaccharides, and here some students they are thinking that, here also there is a mistake done by students. They are thinking that complex polysaccharides means, generally they are thinking that they are heteropolysaccharide. It is not always like that. Please understand. Here complex polysaccharide, whatever they are talking in NCERT, it does not mean that heteropolysaccharides. Here the meaning is something different. So try to understand this. Here they are using a term complex polysaccharide because these are the polysaccharides. These are the polysaccharides having some modified sugars. This is what the meaning is. Till now what we studied: cellulose, starch, inulin, glycogen. These are the four things we have studied. In all these things what we are studying? Glucose only. It is a direct normal glucose only, maybe alpha form or beta form, whatever it is a normal glucose only, that's it. But here what we are talking is, these are modified. Suppose you are talking about the glucose, it is not directly as it is like glucose, it is glucose amine. To the glucose, amino group is attached, that's why it is a modified sugar. Next one, N-acetyl galactosamine. To the galactose, amino group is attached. In this amino, N is present, right? To that N, acetyl is attached. So in this way, it is not directly as it is galactose, it is a modified. So that's why it is N-acetyl galactosamine. So in this way, if modified sugars are present, then they are using a term called complex polysaccharide. Now in this complex polysaccharides also, some may be homopolysaccharides, some may be heteropolysaccharides. What are the standard examples of homopolysaccharide? Chitin. Chitin. So where we have studied about the chitin. So chitin is a polysaccharide. It is a homopolysaccharide made up of same type of unit, and it is generally seen in cell wall of fungi, and also in exoskeleton of arthropods. These are standard examples we have studied. And why it is homopolysaccharide? Because it is having same type of unit. What is that same unit? It is N-acetyl glucosamine. It is N-acetyl glucosamine. Why you are studying chitin under complex polysaccharide? Because it is not just having normal glucose. This glucose is having amino group. In that amino group, to the N, acetyl group is attached. So in this way, it is a modified sugar. Since it is having modified sugar, we are studying under complex polysaccharide. Though it is complex polysaccharide, same N-acetyl glucosamine, it is one unit, the same unit is repeated for several times. That's why chitin is homopolysaccharide. In some books they gave it as heteropolysaccharide, that is wrong. Even some teachers are also teaching that it is heteropolysaccharide, that is wrong. So please correct yourself. N-acetyl glucosamine is a single unit, the same unit is repeated for several times. That's why chitin is example for homopolysaccharide. Please remember this. Clear? These are some important examples from the NCERT. And now if you are clearly observing with the cell chapter, in the cell chapter we studied one term called as galactans. So galactans means they are polymers of galactose. Mannans, they are polymers of mannose. Understood or not? Cellulose term, just now we have studied. Cellulose is polymer of glucose. And hemicellulose and pectin, these two are heteropolysaccharide. Hetero means what? Different types of units are present. So hemicellulose and pectins are examples of heteropolysaccharide. Different types of units are present. So this is one interlinked information, not mentioned in NCERT, but try to remember this. So with this, all these molecules are completed, and now we are going with a different concept that is called as primary metabolites and secondary metabolites. What are primary? Primary means they are compulsory. So for the normal survival of a cell, these are the molecules that are required. So these are compulsory, and you can clearly observe the functions of these molecules within the cell. So till now whatever we have studied, all these molecules are examples for primary metabolites. So they are having the normal visible functions within the cell. That's why they are called primary. But now we are talking about the secondary. What is the role of the secondary in the particular cell? So here, please understand here, still research is going on. The major and important answer is still research is going on to understand the functions of these particular molecules within that particular cell. And in what type of cells we are observing this? Generally, we are observing secondary metabolites in plant cells, fungal cells and microbial cells. Please remember, we are not writing about animal cells because generally these secondary metabolites are not seen in the animal cells. But why they are secondary? Because even though these molecules are removed from the cell, still there is no damage to the cell. That means these are not essential for normal survival of the cell. These molecules are not essential for normal survival of the cell. That's why they are called as secondary metabolites. And for majority of the secondary metabolites, the functions within those cells is not known. Functions within those particular cells is not known. Still research is going on in understanding those things. But majority of these secondary metabolites are useful to human. So there is a big list here. So you need to remember this. So what is the name of this particular component and what is the group and what is the role? Likewise they have given. So observe it carefully. Under the name of pigments, we are talking about carotenoids, anthocyanins and like this. Yes or no? So in this way different are there. And you can understand this, that some of these pigments still in some locations research is going on to understand the specific role. But as an overall, if you are observing, some of these pigments are useful in attracting some of the insects or some birds and some things, right? But still at some locations still function is not known, still research is going on here. Next one, alkaloids. Alkaloids like morphine, codeine. Please remember this one they may ask in the matching or directly they may ask that identify the alkaloid from the given list, like that also they may ask. So you need to compulsorily remember this particular table. Alkaloids examples are morphine, codeine and etc. Terpenoids examples, monoterpenes, diterpenes examples, and essential oils example, lemongrass oil. For the toxins example, abrin and ricin, these are important examples. For the lectins, important example is concanavalin A. And regarding the drugs, vinblastine and curcumin, these are standard examples. For the polymeric substances, so rubber, gums and cellulose. So these are some important secondary metabolites. So these are some standard examples. So at least NCERT examples, please try to remember with corresponding category. Clear? So with that, that's a primary and secondary metabolite concept is completed. Next one is, what is the nature of the bond that is linking these polymers? We are talking about total 3 types of polymers. They are proteins, carbohydrate. Carbohydrate means polysaccharides and then nucleic acid. These are the 3 things. So in the case of protein or polypeptide, what is the bond that is linking 2 units? The bond that is linking 2 units is the peptide bond. So in the peptide bond, what happens? The COOH group of first amino acid and NH2 group of second amino acid are involved, and here due to loss of water molecule, now the peptide or amide bond will be formed. So what is the bond that is connecting two amino acids in a protein? It is the peptide or amide bond. So please understand this. Next one, if we are talking about a polysaccharide, in the case of polysaccharide, many units are attached, many sugar units are attached here, and the bond that is present between these is called as glycosidic bond. It is simply glycosidic bond. Depending on the molecule, this it may be alpha, beta, 1, 4, 1, 2, likewise different are possible. But don't worry about the number, but remember the name of the bond, glycosidic bond. Glycosidic bond is the bond that is present between two sugar units, or else at least one of the molecules should be sugar. Please understand this. Why I am saying like this? So you will understand in the next coming lectures. I will tell you. Please remember to call it as a glycosidic bond. At least one molecule should be the sugar. If sugar is involved in the bonding, that bond is called as glycosidic bond. And here anyhow sugars are present. So that's why here it is called as glycosidic bond. So please remember this. Clear? Next one is regarding the nucleic acid. In the case of nucleic acid, if you are talking about one nucleotide, one unit, within one nucleotide between nitrogenous base and sugar, glycosidic bond. Just now I said, no, if sugar is involved, it is called as glycosidic bond. So here between nitrogenous base and sugar, glycosidic bond is there. Between sugar and a phosphate group, phosphoester bond is there. Between two phosphates, phosphoanhydride bond is present. And these are the bonds that are present within one unit. But our question is, if two units are attached, if two nucleotides are attached to each other, then what is the bond that is connecting here? It is the phosphodiester bond. This is the backbone. So it is the phosphodiester bond that is connecting two adjacent nucleotides. So these are the bonds that are seen within the polymers. And then directly we are going with enzymes concept. So here almost all enzymes are proteins. Please remember in NCERT they said almost all. They did not give all enzymes because there is an exception here. Majority of the enzymes are proteins, but there are some enzymes where RNA is acting as an enzyme. If RNA acts as an enzyme, it is called as ribozyme. What are the standard examples here? Already I have mentioned about these things in the cell chapter also. Yes or no? So in the cell chapter revision also I have mentioned these names. You should compulsorily remember this. 28S rRNA present in 60S subunit of 80S ribosome is having enzyme activity. Next one, 23S rRNA present in 50S subunit of 70S ribosome is also having enzymatic activity. And what is the name of that enzyme activity? Peptidyl transferase. And where it is involved? It is involved in the translation process. Remember these particular examples, and these examples once again we will study under molecular base of inheritance. Under the revision also I will cover these points. When we study about translation, definitely I will tell you about these names. So you should compulsorily remember this. Clear? Next one we are saying that, so since majority, see we are not talking about ribozymes here, we are talking about protein enzymes only. So proteins means they are having primary, secondary, tertiary structures. If they are multi subunit, then only they will have quaternary structure. Yes or no? But whatever it is, until the tertiary it is compulsory. For the majority of these proteins, for their normal biological activities, tertiary structure is compulsory. So that's why they are having different foldings, and in these foldings some gaps may be present, and these gaps are technically called as crevices or the pockets. Crevices are the pockets, and in some of the pockets what happens is a substrate molecule will interact with this. If that particular enzyme is involving in a reaction, substrate molecule will bind to that particular pocket. So the pocket or the crevice where substrate is interacting with the enzyme, that particular location of enzyme is called as active site. Along with the active site, in some cases some extra pockets may be present for regulation of the enzyme activity, and they are called as allosteric sites. But regarding the allosteric sites, they have not mentioned in NCERT. So at least regarding the active site, you please remember. So active site is the location of the enzyme where substrate will interact. At least this point you please remember. So in this way, and next important point is regarding organic catalyst and inorganic catalyst. So in the case of chemistry, we are using the inorganic catalyst like platinum, palladium, all these things. But generally inorganic reactions, they are occurring at a very high temperature and high pressure. But whereas organic reactions in the normal conditions also they are happening. So for the formation of the same product under the chemical reactions in the laboratory, so we may need high temperature and high pressure, but the same reactions inside the organisms can happen at normal temperatures also, normal temperature and normal conditions. So this is what the advantage with biocatalysts. That's why enzymes are called as biocatalysts. Here also they are acting as catalysts, but they are present within the living organisms. That's why they are called as biocatalysts. So that's why enzymes are organic catalysts. They are also called as biocatalysts. So please remember this. Next one is, there are some organisms that are living at very high temperatures, and such type of organisms are called as thermophilic organisms. Since these organisms are naturally present at very high temperatures, definitely enzymes present in them are also active at very high temperature. So that's why these enzymes can withstand high temperatures. Suppose you are taking an enzyme of human. So what is the normal body temperature for the human? It is around 37 degrees centigrade. Suppose you are using human enzyme, so it can withstand up to 40 degrees centigrade. If the temperature is going beyond 40, now it will become inactive. And if the temperature is keep on increased and increased, now the protein will be damaged. Yes or no? But if you are taking an enzyme from thermophilic organism, since it is naturally living at high temperatures, those enzymes can withstand high temperatures and they are still active at high temperatures. One standard example that you need to interlink with biotechnology chapter is Taq DNA polymerase. Yes or no? Under the PCR concept, we studied this. Taq DNA polymerase. So it is isolated from a bacterium that is living at high temperature. That's why it is called as thermophilic bacterium, and its name is Thermus aquaticus. T for thermus, AQ for aquaticus. So from that DNA polymerase was isolated. So that's why it was named as TAQ polymerase, DNA polymerase, and this is involved in recombinant DNA technology, and mainly it is used in PCR technique for the amplification of DNA molecules. Yes or no? So there we are using this. So these are important significances for the enzymes that are isolated from thermophilic organisms. Clear? Next one, they are talking about some chemical reactions. Some reactions are happening by physical process. Generally in these physical process, what happens is breaking of the bonds generally does not occur, simply the change in the state of the matter will occur. So sometimes suppose ice is melting into water and water is becoming vapor. So in these cases, now the bonds are not broken, but simply the change in the state of matter is taking place. But in the case of chemical reactions, what happens is the bonds, old bonds are broken and some new bonds may be formed. And this is what generally happening in the chemical reaction. In the NCERT, they have given one example. Barium hydroxide plus sulfuric acid gives rise to barium sulfate and water. So in this process, what happens? So here initially there are some bonds present here, and these bonds have broken and the new bonds were formed. So that's why it is forming the new product. So in this way, in the chemical reaction, breaking of bonds and making of bonds is taking place. In the same way, another example, starch. Starch is a polysaccharide having many glucose units. When it is hydrolyzed means what? Bond is broken so that it is releasing individual glucose units. So in this case also, what is happening? Bonds are broken, and you can consider this as a chemical reaction. But this chemical reaction is naturally occurring inside the living organisms. This is what we are talking. So in this way, some basic example they give. So when a reaction is taking place, what happens? Some substrates are converted into the products. Substrates are converted into products. So in that case, we can easily measure the rate of reaction. How we will calculate the rate of reaction? The rate of reaction is nothing but amount of products that are formed in unit time. So this is called as rate, and this rate is also called as velocity. When we can call it as velocity? When the direction is specified. For the reaction, if you are fixing a direction, then you can call this particular rate as velocity also. Understood or not? Next one. Here in the NCERT, they are talking that, so there is a significant influence of temperature on rate of reaction. And here what they are saying, for every 10 degrees centigrade change in temperature, the reaction rate may be doubled or it may become half. So what is this particular concept? So I will elaborate this one. So observe this. Generally that sentence is applicable in the range of 10 degrees centigrade to 40 degrees centigrade. In this range it is applicable. Generally that is applicable in this range. What they are saying here, suppose initial temperature is 15 degrees centigrade. For every 10 degrees centigrade increase, 10 degree increase means what? 25 degrees centigrade. Suppose it is another increased by 10 degrees, that means it will become 35 degrees centigrade, suppose. So in this way, from here to here 10 degree increase, from here to here 10 degree centigrade increase. And what they are saying, if 10 degree centigrade increase is happening, and here suppose the rate of reaction is x. Now at 25 degrees centigrade, what is the expected rate of reaction? It will be double. So when the temperature is increased by 10 degrees centigrade, now the rate of reaction will be doubled. At 25 degrees centigrade, if it is 2x, what will be the expected rate of reaction at 35 degrees centigrade? Again, it will be further doubled. So 2x is doubled means it will become 4x. Understood or not? So in this way, if you are observing in this direction, what you are saying, for every 10 degrees centigrade raise in temperature, the rate of reaction will be doubled. Yes or no? This is what they are saying. Suppose you are observing this in opposite direction. For every 10 degrees centigrade decrease or drop in temperature, what happens? Rate of reaction will be reduced by half. So that 4x will become 2x, and here 2x will become x. It will be reduced by half. So in this direction, if you are talking, the rate of reaction will be doubled for every 10 degrees centigrade. In this direction, if you are observing, the rate of reaction will be decreased by half for every 10 degrees centigrade decrease in temperature. This is what the meaning of this particular sentence. Clear? So this is what. So observe this carefully. Okay. Next one they are talking about the difference between uncatalyzed reaction and catalyzed reaction. Uncatalyzed reaction means catalyst is not present. If catalyst is not present, the reaction will occur very slowly. So if catalyst is present, the reaction will occur very quickly. So for that they have taken one example. So in both the cases, it is the formation of carbonic acid. It is the formation of carbonic acid, H2CO3. So how the H2CO3 will be formed? CO2 plus H2O will give rise to H2CO3 carbonic acid. For the same reaction, if catalyst is not present, what they are saying here? 200 molecules will be formed in 1 hour. You are taking the water, and in that water you are sending the CO2 and leave it like that. Don't add any enzyme. Then what happens? And here 200 molecules of carbonic acid will be formed in 1 hour. 1 hour means 60 minutes. Seconds, how many seconds? 60 into 60 seconds. Yes or no? So that much time it is taking for just 200 molecules. But in the same thing, if enzyme is present. So what is the enzyme that is involved here? Carbonic anhydrase. If the enzyme is present, then what they are saying? 6 lakh molecules are formed in 1 second. In every second, these many are produced. In 1 second, 6 lakh are produced. Here, in 1 hour, means 3,600 seconds. For that, just 200 molecules are produced. Now you can understand how fast they are happening in presence of enzyme. So this is the power of enzymes. This is what the example that they are saying here. Clear? So in this way, here the rate of reaction is accelerated, increased by almost 10 million times. Almost 10 million times. This is what one example given in NCERT. So please to try to remember this. And then finally they are saying that in this way many reactions are taking place inside the organisms. So in the living organisms, all the reactions are occurring in presence of enzymes only. There are no reactions that are occurring without the involvement of enzymes. This is what they are saying. And next one, the conversion of glucose to pyruvic acid is a 10-step reaction, and this all 10 steps are commonly called as glycolysis pathway. Already you studied about this particular pathway in respiration in higher plants chapter. Clear? But though you are studying in higher plants only, but that reaction is applicable even for animals also. So please remember this. And here what happens? Some sequence of reactions are taking place. And this set of sequence of reactions is generally considered as metabolic pathway. Clear? And now this pyruvic acid may have again different fates. So in the case of oxygen conditions, if oxygen aerobic conditions, if oxygen is available, now this pyruvic acid can be converted into acetyl coenzyme A, and now it can enter into the Krebs cycle. So that is one pathway. If oxygen is not present in different organisms, different other fates are there. If you are talking about the human skeletal muscle cells, in the human skeletal muscle cells, what happens? This pyruvic acid will be converted into lactic acid. And not only in human skeletal muscle cell, in lactic acid producing bacteria, LAB, lactic acid producing bacteria. Regarding this, we have studied in microbes in human welfare. So remember this. In that bacteria also, pyruvic acid will be converted to lactic acid. Suppose you are talking about the yeast that is called Saccharomyces cerevisiae. This also we have studied once again in microbes in human welfare. And there what happens? From the pyruvic acid, ethanol will be produced, ethyl alcohol. Yes or no? Ethyl alcohol will be produced. So in this way, in different organisms, in different conditions, so different products may be produced. So in this way, different metabolic pathways will lead to different products. This is what they are saying. And next one, they are saying that how exactly these enzymes can bring about these higher rates of reactions. If enzyme is not present, reaction is occurring, but it is occurring very slowly. If enzyme is present, the same reaction is occurring very quickly. So how these enzymes are responsible for this quickness? So this is what they are taking here. Suppose substrates are converted into products. In this particular process, what exactly happens here is, so substrates now generally they are interacting with this enzyme. First substrate will interact with the enzyme. So now what happens here? Some changes that are taking place here. But ultimately, from this particular diagram, you can easily understand that if enzyme is not present, the energy required to pass the transition state is very high. Please remember this, transition state is present in both the cases, whether enzyme is not present or enzyme is present, whatever it is, transition state is compulsory. But what is the amount of energy required to pass that transition state? That is very important. If enzyme is not present, more energy is required to pass this transition state. If enzyme is required, what the enzyme will do? Enzyme will decrease the activation energy. So that's why less energy is enough to pass that transition state, because enzyme is decreasing the activation energy. Please understand this. Enzyme is not changing the energy of substrates. Enzyme is not changing the energy of products. Then what enzyme is doing? Enzyme is decreasing the activation energy. When the activation energy is decreased, now more substrates can easily pass through the transition state, and then they can cross this activation energy, and then they can easily form the product. In this way, enzymes are responsible for more rate of reaction. This is what they are saying here. Simple concept. Clear? So this is what they are talking here. So in this process, what happens? First substrate will interact with the enzyme, so that enzyme substrate complex will be produced, and now this slowly some intermediate forms will be formed. Some breakage of old bonds, formation of new bonds, and this also some sequentially some products will be formed. Yes or no? So and all these things are called as altered states. Altered states are seen in the transition state, and these altered states are also called as reaction intermediates. Generally in the organic chemistry you will study that there are some substrates and products. In between this there are some reaction intermediates. Those intermediates are seen in the transition state. At the transition state we are observing reaction intermediates. Intermediate forms we are observing, altered states we are observing. But please remember, since transition state energy is very high, energy is very high means stability is very low, stability is very less. Yes or no? So that's why these reaction intermediates, since they are having very high energy, they are highly unstable. They are highly unstable. That's why quickly they are changing their form, and then they are converted into stable products. So substrates are stable, products are stable, but the transition state, intermediate states are not stable. These are some important points that you need to understand from this explanation of NCERT. So if on the x-axis, if you are observing this reaction progress of the reaction or reaction progress, on the y-axis they are mentioning the potential energy. So this is how we are observing the diagram. Is it clear? The same thing they have mentioned here. You can go through NCERT explanation, and more and clear explanation I gave in regular lecture also. You can go through that one also. Clear? And next one is regarding another two names. Exothermic reaction, it is also called as spontaneous reaction. And other one is endothermic reaction, it is also called as energy requiring reaction. Observe this. In the endothermic reaction, energy is needed, but in the exothermic reaction, energy is released. Now, the important thing is the diagram. How exactly you will represent this particular one in the form of energies? Observe carefully. First, we will talk about exothermic reaction. In the exothermic, exo means what? Outside. Energy is released outside. Why energy is released? Because the energy of substrates is higher when compared with the energy of products. This is the energy, right? So energy of substrate is higher when compared with energy of product. So that's why when substrates are converted into products, then what happens? Energy is released. That's why these reactions are called as exothermic reactions. In the exothermic reaction, suppose this is the transition state without enzyme. In presence of enzyme, let us draw another one. Suppose it is like this, like this. So in this case, what happens? Energy of substrates and products remains same. But what the enzyme is doing? Enzyme is decreasing the activation energy. Enzyme is decreasing activation energy with enzyme. With enzyme. This is without enzyme. This is without enzyme. Observe carefully. This is how you will get the diagram for exothermic reaction. And you should compulsorily know the reaction diagram for endothermic. Endo is inside. That means energy is taken inside. So this energy is generally provided in the form of heating or through some other process. Now what you need to do? Here also let us make substrate energy like this. Energy of substrate here. But here what is the energy of the products? Energy of the products is more than energy of substrate. That means suppose this is the line, you need to draw the product above. Energy of products is higher when compared with energy of substrates. So from where you will get this excess energy? Thermic, by heating, by temperature. From the temperature, by heating you are getting this energy. And in this case also the transition state energy is higher. Observe carefully. Transition state energy is higher. And here if enzyme is present, it will lower the activation energy. It will lower the activation energy. So now observe carefully. So whatever is the condition, so here whether it is exothermic or endothermic, whatever it is, in both the cases energy of transition state is higher. energy of transition state is higher than energy of substrates and energy of products. Understood or not? In both the cases, the energy of transition state is higher than the energy of both substrates and products. Very, very important. So this is how the diagram will be changed. So in the NCERT they gave this type of diagram. So the NCERT diagram will correspond to exothermic. Suppose they are talking about endothermic. This is how we need to draw the diagram. So in the regular lecture also I have clearly explained. So please go through this particular one. Clear? And next one we are talking about nature of the enzyme action. In the enzyme action, what happens? First, enzyme and substrate will interact with each other so that they are forming the enzyme substrate complex. So once this enzyme substrate complex is formed, now what happens? Breakage of some old bonds will occur and then formation of some new bonds will occur. Once the new bonds are formed and all these intermediates are crossed, and finally it will result into the product. So now this product is interacted with enzyme, so that's why it is forming enzyme product complex. And now from this product will be released, and now this enzyme will be originally formed, and now this formed enzyme will once again use it for another cycle. So that's why in the catalytic cycle of an enzyme, in the NCERT they have given 4 steps. So observe carefully. Substrate will interact with active site of the enzyme, and then what happens? Now it will induce the enzyme to alter its shape so that it will fit more tightly with the substrate. And then what happens? Breakage of the chemical bonds, and then finally formation of enzyme product complex. When the enzyme product complex is formed, now what happens? Now product is not exactly fitting, now it will be released and enzyme will get back its original shape, so that now the products are released and the free enzyme will be again reused for another cycle. So in this way, now it will be cyclically used in several rounds of these chemical reactions. So this is what about the catalytic cycle of an enzyme. With that, that concept is completed. Next we are studying is what are the various factors that are affecting the enzymatic activity. Please remember, generally the enzyme is present in its tertiary or quaternary structure based on the number of subunits. And please remember, some factors they can affect this tertiary or quaternary structure. So the factors that are affecting this tertiary or quaternary structures, so they will affect the enzyme activity. So in this we are studying some important examples like temperature, pH, substrate concentration, and then sometimes binding of some molecules that are regulating enzyme activity like inhibitors. So these are some standard examples that we are studying here. Now let us quickly go through this particular one. So regarding the temperature and pH, if you are observing, what is the effect of pH here? For every enzyme at a particular pH, the maximum activity is seen. At a particular pH value, the maximum activity will be seen. And if pH is increased or if pH is decreased, now what happens to the activity? Activity decreases. So that's why the pH value at which maximum activity is seen for the enzyme, that pH value is called as optimum pH for that particular enzyme. So in this way, different enzymes will have different optimum pH values. Yes or no? Some simple examples, even though the digestion and absorption chapter is deleted. So now let us see this. In the case of saliva, for the enzyme called ptyalin or salivary amylase, the optimum pH required is 6.8. So, and this saliva when mixed with the food, it is forming the bolus. When you are swallowing the bolus, it will go into the stomach. But in the stomach, what is the pH? Approximately 1.8. But for the salivary amylase, optimum pH is 6.8. Now the saliva is going along with the food into the stomach, but in the stomach the pH is 1.8. Now is it active at that temperature? Is it active at that pH value? No. So in this way, for every enzyme there is a specific pH that is required. So that pH where enzyme is having maximum activity is called as optimum pH. So in this way, enzymes present at different locations, they are active at different pH values. So that's why they are called as optimum pH values for those particular enzymes. This is what the concept of pH and the effect of pH on enzyme activity. Same way for the temperature also. For every enzyme at a particular temperature, they will show the maximum activity. If the temperature is decreased further or temperature is increased further, in both the cases, enzyme activity will be decreased. Now let us take simple example of the human. In the human, the enzymes will show the maximum activity at 37 degrees centigrade. Let us say that suppose I got a fever. So I got fever means what happens? Body temperature is increased. When the temperature is increased, now what happens? Enzyme activity will be decreased. In that case, enzyme activity will be decreased. Yes or no? Now let us take another example. Let us take some bacterial paste, and let us take the E. coli bacterium. For the E. coli bacterium also, optimum temperature is 37 degrees centigrade. If I am keeping them at 37 degrees centigrade, they are growing very nicely because all the enzymes are working effectively. Suppose I am taking this microbial paste and I am keeping it 50 degrees centigrade. Now what happens? Now the enzyme activity will be lost and they may die because it is very high temperature. Suppose I am keeping this in the refrigerator at 4 degrees centigrade. Now what happens? Temperature is decreased, enzyme activity will decrease. Now the growth will be decreased. So in this way, for the enzymes also, for the optimal activity, for the optimum activity, for the maximum activity, there is a required temperature. If the temperature is reduced or increased when compared with optimum activity, now the activity will be decreased. So in this way, there is optimum temperature, there is optimum pH for the activity of these enzymes. Clear? In the NCRT, with respect to temperature, they have added one more sentence. The sentence is, if the temperature is increased to some extent, organism will survive. But if temperature is keep on increased, organism may die. We will talk with respect to the enzyme. So if for the enzyme specifically, if you are doing in the laboratory, in a tube you are doing, and you are keep on increasing temperature, up to a particular temperature, it may withstand. Enzyme activity will decrease after the optimal temperature. But if you are keep on increasing the temperature, the protein may be permanently damaged. That is the problem with increase in temperature. But decrease in temperature, generally it is not causing damage to the protein structure, but activity will be decreased and it will restore or it will protect and it will prolong the enzyme structure. So in this way, so that's why generally if you want to store this particular protein, we are keeping it lesser temperature so that we can store it for longer duration, and in future when you are bringing it back to the normal temperature, again it will be functional. So in this way, for storing of the proteins, we are preferring this lower temperatures. This point also they have mentioned in NCRT, so please go through this one. Next one, concentration of the substrate on enzyme activity. So here generally what happens, if very less concentration, we are starting with very less concentration, then what happens, with increase in substrate concentration, enzyme activity is also increasing. In this way, for to some extent it is keep on going like this. But once a saturation is happening, further increase in substrate concentration will not cause further increase in activity. From here onwards you will get the straight line. So that means enzyme activity or rate of reaction or velocity of reaction is increased up to a certain extent of increase in substrate concentration, and after that, even though substrate concentration is increased, still the enzyme activity or the rate of reaction will remain same. This is what the standard graph that we are getting. How exactly we are getting this? Everything I have explained clearly in the regular lecture. So please go through that if you want more details. And once you are making the standard graph, then what you are doing? Whatever is the maximum activity you are getting, that is called as Vmax, maximum velocity or maximum rate of reaction. On the graph paper, you are making this. Whatever the value you are getting here, take off of this. And from this point, you just draw one line onto this particular graph line. And wherever it is cutting, from that point you just make one straight line onto this x-axis wherever it is touching, and that is called as Km. Then what is this Km? Km is nothing but it is one substrate concentration value. Km is nothing but one substrate concentration value at which half of the maximum velocity. Vmax by 2 means what? It is the half of the maximum velocity. So what is Km value? It is the substrate concentration at which half of the maximum velocity is observed. It is called as Km. It is also called as Michaelis-Menten constant. Clear? How exactly we are making this graph? Very clearly I have explained in the regular lecture. So those who are having still doubt on this particular graph preparation and what is the role of Km, if you are having this type of doubts, please go through the regular lecture. Clear? Next one, we are talking about the inhibitor. These are some chemical molecules and different inhibitors are acting in a different way. But in NCRT they are talking only about the competitive inhibitor. So I am also talking only about the competitive inhibitor. So what is the action of this competitive inhibitor? competitive inhibitor is inhibiting the enzyme activity. Ultimately it is inhibiting enzyme activity, but how exactly it is inhibiting? By competing with substrate, Why it is competing with substrate? Because some portion of this inhibitor is having structural similarity with substrate. For the substrate is binding to the enzyme at active site, right? So for that substrate, whatever the portion that is interacting with the enzyme, similar structure is seen in competitive inhibitor also. That's why it is also able to interact with same enzyme at the same active site. That means both substrate and competitive inhibitor, both of them are trying to interact with enzyme at the same active site. So what happens? If now there is a competition between these two. Now in the competition, who will win? Those which are having higher concentration, definitely they will get the chance to bind with enzyme. So that's why there is a graph preparation here, but in NCERT they have not given about the graph preparation, but how exactly the graph will be changed, why Km is increased in the case of competitive inhibition, okay, but Vmax will remain same. These concepts very clearly I have explained in the regular lecture, and here it will take a little bit more time, that's why I don't want to go into that, but I have explained very clearly in the regular lecture. Those who are still having doubt on this particular graphs preparation, so you need to go through that regular lecture. Clear? So in this way what happens? Competitive inhibitor is competing with the substrate to bind with active site of the enzyme. So in this way, if inhibitor is binding with enzyme, enzyme cannot form the product. But if substrate is binding, yes, it can form the product. So how we can release the competitive inhibition? By increasing the substrate concentration. This mechanism also I have explained very clearly in regular lecture. So please go through it. And here in NCERT they gave one example. What is this? Malonate. Malonate, also called as a malonic acid is acting as competitive inhibitor, and it is competing with substrate. What is the substrate? It is succinate or succinic acid. It is competing with succinic acid or succinate for binding with the enzyme, that is succinate dehydrogenase. For succinate dehydrogenase, standard substrate is succinate or succinic acid. But here who is coming here? Malonate is also trying to bind with succinate dehydrogenase. Why malonate is binding here? Because malonate is having structural similarity or resemblance with succinic acid. This is what they are talking here. So in this way it is acting as competitive inhibitor. Generally these competitive inhibitors are used in controlling the bacterial pathogens. That means we can use them as antibiotics. That's what the meaning is. Next one, classification and nomenclature of the enzymes. So here total 6 classes of enzymes are present. Again in each class, again 4 to 13 subclasses are present. Based on the size of the class, different subclasses are present. And Enzyme Commission, EC, Enzyme Commission has given 4-digit number. Out of these 4 digits, first digit will represent the class, second one will represent the subclass, third one represent the subclass, and the last one will represent the serial number. And these points also very clearly I have explained in the regular lecture. If you still have the doubt, so please go through that. And now what are these 6 classes? You need to remember these 6 classes in the same sequence. What are these 6 classes? O, T, H, L, I, L. These are the first letters of all these 6 classes. Oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases. You should remember in the same sequence. Because class number 1 will be oxidoreductases. Class number 2 is transferases. Likewise, in the same sequence you need to talk. First we will talk about the class number 1. These are called as oxidoreductases, also called as dehydrogenases. Here what happens? One substrate will be oxidized, other will be reduced. So in this way these reactions are taking place. In the case of transferases, some groups are transferred from one substrate to other substrate so that different products are formed. This is what about the transferases. Next one is regarding hydrolases. These enzymes, they are breaking the bonds with the involvement of water molecule. So in this way they are breaking different types of bonds. Next one, lyases, and these here what they are happening is they are removing some of the groups without the involvement of water molecule, and generally here we are observing carbon-carbon double bond formation at these locations, and those enzymes are called as lyases. Next one is isomerases. These enzymes are involving in conversion of one form of substrate into other one, and generally these are the isomers, different isomers. They may be optical isomers, positional isomers, or geometrical isomers. Different types of isomers may be formed because of these enzymes. And the last one is ligases. Ligation means what? Attachment. So that means they are linking different molecules so that they are forming different products. So generally they are involving in joining of different types of molecules. This is what they are talking. So these are the 6 classes. Please remember, you need to remember these classes in the same sequence only. Clear? And finally we are talking about the cofactor concept. So here, before directly going with the cofactor, first we need to talk about holoenzyme or conjugate enzyme. This is one type, and the other one is called simple enzyme. What is simple enzyme? In the case of simple enzyme, protein part will be there, and that protein part itself will act as an enzyme. That's it. In the case of simple enzyme, I'm repeating, in the case of simple enzyme, protein part is there. That protein part itself will involve in the catalysis reaction. And in the case of simple enzyme, there is no non-protein part. Only protein part is there, that itself will act as an enzyme. But in the case of holoenzyme or conjugate enzyme, there is a protein part, and along with the protein part, there is a non-protein part. So please remember. So in this case, what happens? This protein part is called a poenzyme because this protein part on its own, it cannot do the catalysis. It cannot do the catalysis. This protein part should compulsorily interact with non-protein part called as a cofactor. If cofactor is not present, a poenzyme cannot do the reaction. Please remember this. But in the simple enzyme, this cofactor concept is not there. Protein part is there, that itself will do the reaction. But in this case, the protein part is called a poenzyme because it cannot do the catalysis without the involvement of cofactor. That's why in the case of polar conjugate enzyme, non-protein part called cofactor is very important. And these cofactors are 3 types. The one is called as phosphate group, next one is coenzyme, and the last one is called as metal ion. But broadly, if you are classifying these cofactors, some are organic cofactors, some are inorganic cofactors. Inorganic cofactors are called as metal ion cofactors, whereas organic cofactors are again 2 types. Some of these organic cofactors are tightly attached with a poenzyme. Tightly attached with a poenzyme. What is the meaning of this? Always they are attached with a poenzyme, and these are called as phosphate groups. Whereas some organic cofactors, they are transiently interacted with a poenzyme. What is the meaning of this? Only during the chemical reaction they will come and attach, they will do the reaction. Once reaction is completed, they will be released. So that's why they are saying that transiently means temporarily associated with a poenzyme. That means only during the reaction they will come. They will complete the reaction. Once reaction is completed, they will be released. And that type of cofactors are called as coenzymes. That's why we say that phosphate groups are tightly associated with a coenzyme, and coenzymes are loosely associated or transiently associated with a coenzyme. This is what we are saying. So overall, 3 types: phosphate group, coenzyme, and metal ion cofactors. These are the 3 types of cofactors that we are studying here. Now some examples also they gave. For the phosphate group in NCERT they gave an example. Heme is the phosphate group for the enzyme that is called as peroxidase or catalase. And what this peroxidase or catalase will do? They will cause the breakdown of H2O2 into H2O and water. This is what the function of these enzymes. Next one, coenzyme. What is the example of coenzyme? NAD, NADP, FAD, FMN. NAD means nicotinamide adenine dinucleotide. NADP is nicotinamide adenine dinucleotide phosphate. FAD, flavin adenine dinucleotide. FMN, flavin mononucleotide. These are various examples of the coenzymes. Generally, these coenzymes are derived from some different types of vitamins. Okay, and these are involved in various enzymatic reactions. Next one, metal ion cofactor. So zinc Zn+2 is the cofactor, metal ion cofactor for the activity of the enzyme carboxypeptidase. What is the meaning of this carboxypeptidase? What it will do? It is a peptidase, a protein or the peptide breaking enzyme. And from which end it is breaking? From the carboxy, from the C-terminus end it is breaking. So carboxypeptidase is the enzyme that is breaking amino acids from the protein starting from C-terminus. That means from the C-terminus it is going towards N-terminus. Please remember, it is the breakdown, not the synthesis. Observe carefully. So these are the various examples for the different types of cofactors. With this, the NCERT content for the biomolecules is completed. Clear? So before concluding this, once again I am telling you, revision lectures are available on both Live Prep app and also on our YouTube channel, but the test series can be given only through the app. Clear? So with this is completed. So in the next lecture, we will meet with a different chapter. Thank you.